Thank you very much for the kind introduction. I'm happy to be here. I'm not working on Huntington's disease, as Patrick just mentioned, but definitely this work has been inspired by some of the findings in Huntington's disease, and I hope that I can maybe give something back to you as a community, but we will see. As Patrick mentioned, I'm mainly working on C9ORF, ALS, and FTD. So since this is a Huntington conference, maybe a brief introduction to ALS, amyotrophic lateral sclerosis, a severe motor neuron disease that typically leads to death within 2 to 5 years after onset. Here both the upper and lower motor neurons degenerate, and this leads to progressive muscle weakness and hyperreflexes and spasticity. Frontotemporal dementia is a kind of heterogeneous disease where, depending on which areas of the frontal or temporal lobe are degenerating first, it can start with impairment of speech and language, or alter the behavior of the patients profoundly. And these diseases are linked together through genetics and pathology. So about half of the FTD patients have inclusions of the protein called TDP43, and 90% of all ALS cases also have inclusions of this protein. And I'm working on the most common mutation that is causing both of these diseases. So in people of European descent, about 5 to 10% of ALS and FTD patients have a massive expansion of a G4C2 repeat in the first intron of this previously uncharacterized gene, C9ORF72. And so this is very different from Huntington's disease. Here, the healthy individuals typically have 2 to 3 repeats, and patients have almost all patients have hundreds of repeats. So there's no clear defined cutoff as in Huntington's disease. When this mutation was discovered, the first hypothesis was that because the RNA actually kind of is sequestered into nuclear foci, that this could sequester RNA binding proteins like the muscle blind proteins in myotonic dystrophy. And a number of proteins interacting with this RNA have been identified, but there's no muscle blind of C9 so far. And the second thing that was discovered was that the repeat inhibits the transcription of the mutant allele. So now we know that this can cause microglia dysfunction and also regulates autophagy. So this is another pathomechanism, but it cannot explain the profound pathology in these patients. So as I mentioned, these patients have TDP43 pathology like most ALS patients and half of the FTD patients. But in addition, they have aggregates that were initially identified by P62 staining, but couldn't be were TDP43 negative. So my hypothesis was, after I heard a talk from Laura Wainum about run translation in myotonic dystrophy, that this intronic repeat could also be translated into repeat proteins. And if you look at this in a kind of a DNA program, there could be 3 different repeats that we call dipeptide repeat proteins. And in fact, all 3 of them are expressed. So this project started kind of as a mission impossible and has been like that all the way. So I'm very confident it will have a happy ending. So in the beginning, it's obvious that the gene is silenced, introns are not translated, and there's actually no start code on any reading frame. So these DPR proteins should never be made. Then when we actually detected them, a pharma guy told me that I killed C9 as a therapeutic target because you cannot fix the ribosome. Then also pathologists claim that DPRs are just innocent bystanders because they don't correlate very well with neurodegeneration. And at the beginning of the project, actually, many companies told me that it's impossible to make antibodies against these proteins because especially the glycine alanine is so hydrophobic that it just wouldn't work. actually we got beautiful antibodies, and then we thought, okay, let's do immunotherapy against this, but this should also not work because it's an intracellular antigen, of course. Now it looks promising, and now people tell me you can't bring it to the clinic. So I'm at this last step now, but I'm still confident. So let's go back to the beginning a little bit. So we were actually able to generate antibodies against all of these DPR proteins, and they specifically detect inclusions in C9 cases, but not in other ALS and FTD patients. And these inclusions are in fact P62 positive and TDP43 negative. So they are really these mysterious inclusions that we wanted to discover or elucidate. And poly-GA is by far the most abundant one. Essentially all TDP43 negative inclusions are GA positive. Later on, we found that also the antisense transcript is actually expressed and is translated into very rare additional DPR proteins. And now we made beautiful monoclonal antibodies that really detect nothing in control tissue and abundant inclusion pathology in these patients. And we'd like to use these for therapeutic studies. So I already mentioned that actually there shouldn't be any DPR proteins. So, but actually it does work even in cell culture. If you express repeat of increasing length, in this case as an exon in HEK cells, you start seeing around 40 repeats, you start seeing expression of these DPR proteins. So I think now it has been convincingly shown that the repeat expansion kind of causes intron retention, so then this solves the intron translation problem. And it seems that actually the ribosome is a lot less picky than people think from their textbook knowledge. So actually many codons can lead to some sort of translation, and the CUG that's in the GA reading frame is a relatively good start codon. Also, I think that these proteins are extremely stable, and that's why even low-level expression leads to gradual accumulation of these proteins in the brain of patients. And all this seems to be kind of triggered by stress, and there are some signaling cascades also worked out how this could potentially happen. So one translation was initially discovered by Laura Rainum in CAG repeats, actually in SCAR8 and in myotonic dystrophy type 1. And then we and others later also showed it in C9. And now it has been shown for a number of repeat expansion diseases, including Huntington's disease, where there is translation in the other reading frames. So it would be cool to actually develop a specific inhibitor for this mechanism, and there are a couple of candidates that have been reported, but they have usually a relatively narrow specificity. So they tend to inhibit global translation, and of course that's nothing that you would want to give an actual patient. So as I said, I don't work on Huntington's disease, but I found this work by Laura Rainum convincing that she generated antibodies against the other reading frames of the CAG repeat, especially the C-terminal peptide that then leads to a stop codon relatively quickly. And this gave her beautiful antibodies that stain inclusion pathology in addition to the polyQ in Huntington cases. And this seems to be especially prominent in juvenile Huntington cases with a large number of repeats, where there's widespread pathology of these other translation products. And recently she also, in a collaboration, could show that these proteins are expressed in a mouse model with a pure CAG repeat expansion, at least in aged mice. Okay, so back to C9. So we generated multiple cell models and animal models, where we made synthetic genes that express individual DPR proteins, because this is a lot more stable to maintain than the original repeat. And especially the transfection of GA repeats leads to nice inclusion pathology in the cytoplasm and also in some cells in the nucleus, which nicely mimic the inclusion pathology in patients. We mostly work in lentivirally transduced rat primary neurons, and one of the reviewers asked us, how much is this overexpressed? And in fact, it's not that much overexpressed. So the inclusions look very similar to in size to the patient inclusions, and they're even less intense than the patient inclusions. So I think we are at a relatively physiological level, actually. So this part is actually inspired by Huntington's disease. I'm sure you have seen that paper by Ruben Fernandez Busnadiego, who is also, or he was in Munich at that time. And we thought we also want to see how the fibrils of poly-GA look like. Is it an amyloid or not? And in fact, the fibrils are kind of disappointing. From the resolution, we cannot really tell the structure. So it's compatible with an amyloid, but it didn't really bring anything new to the table. But what it really, what was striking was the enrichment of proteasomes here labeled in green, that there's like a 30-fold enrichment of proteasomes in these inclusions. And you can already see that many of them are very close to the fibrils or actually ribbons in these inclusions. Because cryo-electron tomography is a single particle method, we were able to average the particles, and then later the computer spit out essentially 4 different structures, 2 very similar ground state structures and 2 substrate processing structures. So the proteasome is a molecular machine that kind of wiggles around while it processes its substrates, much like the sink disposal machines in maybe in US kitchens. And we found that close to the aggregates, there's actually a very large increase in this substrate processing state. That sounds good, so substrate processing, but in this case, it actually means that the proteasome is stuck. So if that wiggling stops, there is no protein degradation anymore. So it looks like the proteasome is actively chewing on these aggregates, but is choking in the process. We also could show that the proteostasis is impaired because a reporter protein is highly upregulated in cells the more that express more and more poly-GA. And as I said, this was initially found by, this method was discovered by Ruben Fernandez, and we later also collaborated on TDP aggregates, also in ALS, that are kind of amorphous in our hands, and they also show enrichment of these proteasomes. So we think that this is something specific to ALS, because in the polyQ exon 1 inclusions that he analyzed first, there is actually no enrichment of proteasomes, and instead it messes with the ER structure in these inclusions. The alpha-synuclein, later they did also look at alpha-synuclein inclusions, and yet again they look different. So it's really critical to look at all of these inclusion pathologies individually. It's not just like a piece of junk in the cell. So this analysis can really tell us something on the cell biology, I think. Okay, so aggregating proteins are often transmitted from cell to cell. Most famously, this has been shown for tau. So we looked whether DPR proteins also can be transmitted from cell to cell in a co-culture assay with green aggregates and red labeled receiver cells, and then we quantified or we analyzed the yellow cells that took up the inclusions by FACS, and we can show that there's especially the hydrophobic DPR proteins, GA, GP, and PA are nicely transmitted from cell to cell. We have used another system to analyze this. We have G4C2 constructs that are tagged in all reading frames, and we treated these cells with brain extracts from C9 patients. And here we can show that compared to a control extract, the C9 extract really increases the aggregation of this construct, or the protein derived from this construct in our cells. We think this stimulates translation of the RNA, and it also promotes aggregation and then stabilizes the stuff. We have also shown this in a FACS-based assay, where we this cell-to-cell transmission also kind of comes with the promise that you might be able to interfere with an antibody with this process. So this is what we tested here in this FACS-based assay. So we essentially quantified this GA from the reporter using intracellular staining, and again the C9 extract significantly increased the levels of GA produced in these cells, or aggregating in these cells, while when we pre-incubated our patient extracts with our monoclonal GA antibody, this was no longer the case. So we think we can come up with this model that GA can be transmitted from cell to cell and leads to kind of seeding of new aggregates in receiver cells, and this process can be potentially blocked by monoclonal antibodies. And later I will also show you that this causes actually non-cell autonomous effects in the receiving cells. So I mentioned that these inclusions have impaired proteasome function, and then we thought maybe this is the link between the C9 mutation and the TDP43 pathology in these patients, because proteasome inhibitors are known to induce TDP43 aggregation. So in cell culture, that's in our system, it leads to this small punctae. TDP is normally a nuclear protein, and now there are small cytoplasmic dots, essentially. This can be is nicely replicated when we express poly-GA. Then we also get these dots even in the absence of the proteasome inhibitor. So this nicely mimics the effect. And then there's, in the past talk we heard about PDE. There's a PDE4 inhibitor, Rolipram, that is known to stimulate proteasome activity. And this indeed can rescue this effect. So here we have virtually none of these cytoplasmic TDP puncta, which can also be quantified in this automated pipeline. So, and we have shown that this also works in neighboring cells. And this is all in this paper. This is kind of a complicated story, so I only showed like the key figures out of this. And then we again tested whether antibodies can interfere with this critical pathway. And indeed, when we treat our cells with a GA antibody, these include the cytoplasmic mislocalization of TDP induced by GA goes away. So, and also the antibody leads to a reduced expression of these aggregate proteins. So it seems that even there is some sort of intracellular effect, but it also happens in a receiver, donor-receiver assay. Here I show you only that like the EC50 of our antibodies is in the nanomolar range, so that's kind of nice. So why do I think this is a good target for immunotherapy? Because GA, we think that GA really links the C9 mutation to the TDP43 pathology that probably ultimately kills the neurons. Poly-GA is, unlike all these other aggregating proteins, only found in patients. So it has no endogenous function, and messing with it should be safer than messing with tau or endogenous Huntington function. And also, unlike poly-Q, poly-GA stretches are not common in the proteome. So we think this could be a safe target. So now we developed a mouse model that expresses poly-GA, and these mice have a motor deficit. So the wild-type mice nicely walk across the stick in a few seconds, and the transgenic mice walk a little bit funny, and they take either a long time to cross the stick, or they will fall down eventually. So this is an assay that you can easily perform and repeat longitudinally. These mice develop inclusions in the spinal cord and brainstem around the age of 4 months, or 3 to 4 months. And now we thought we can't afford making that much antibody to do a passive antibody therapy. So we thought we will just immunize our mice against GA. And here we used a a conjugate of ovalbumin and GA10 to make GA more immunogenic. This is also how we made our monoclonal antibodies. So we just essentially copied that protocol. In addition, we used also GA15 because in the Alzheimer field, people found that A-beta-42 is without any carrier highly immunogenic. So we thought maybe this particulate aggregates could also induce an immune response by themselves, but actually they didn't. So there was actually no immune response against this GA15, while the GA10 coupled to ovalbumin induces extremely high antibody titers. So the company guys were right. GA is not immunogenic, but you can make it immunogenic by conjugating it to other stuff. So in the other slides, you will always see that I keep GA15, but essentially it serves as an additional negative control because it has no antibodies and has also no effect in any of the tests. So the mice, as I said, developed aggregates around 3 to 4 months of age, and this is also when they develop motor deficits. So in this beam walk assay, the wild type mice take about 4 seconds to cross the stick, and this is pretty much constant over time. While the transgenic mice that were immunized with PBS only developed this deficit, and the GA15 immunized mice without antibody response also developed these deficits. The group that actually got an antibody response, these ovalbumin GA10 mice, they had a nicely improved motor function, although we didn't quite reach wild type level. So in this case, we immunized before the onset of symptoms, so that because it takes a while to induce a robust antibody titer, but we have done it also. I'll show you the data later, where we started here and here with the immunization. First, we wanted to see if this can actually reduce GA levels, and there is an effect, but it's relatively modest, I would say. There is also an effect on the secondary TDP-43 pathology. So also the mice show some cytoplasmic mislocalization of TDP that is largely rescued by the ovalbumin GA10 immunogen. But actually the most striking effect is on inflammation. So we found that the transgenic mice have a massive microglia activation that is largely prevented by ovalbumin GA10 immunization. So I think this is actually the key effect that is elicited by our immunization. And we heard so much about neurofilaments by Henrik's beautiful talk. And we, of course, also measured neurofilaments. And indeed, there is a reduction of about 50% in our preventive study. So I think this could be potentially meaningful if this would be replicated in patients. But of course, most patients are already symptomatic. And then we repeated the study in symptomatic mice, starting at the age of onset, essentially. And then we, with 2 different antigens, we kind of replicated this reduction of neurofilament light in the CSF. And then we got more bold and started even later. And this is a little bit messy because now we switch to blood, but we still see this 50% reduction. And in this case, we talked to regulatory people how to bring this to a patient. And they told us that our vaccine is too aggregation prone, so we can't make this into a drug. They told us that we have to optimize it to be more soluble. And this is actually this SVAX formulation. I can't tell you what it is now, but it is far more soluble than the original antigen. It's still antigenic, and it still reduces neurofilament light. So I think our model right now is like this, that GA inclusions are not only toxic to the cells that initially make them, but they can also be transmitted from cell to cell and induce some toxicity in neighboring cells, especially we have shown that for proteasome inhibition and TDP-43 mislocalization. And based on this vaccination data, we postulate that transmitted GA also activates microglia and kind of stimulates a neurotoxic phenotype in them that contributes to neurodegeneration in our mouse model. So that's why we think that GA antibody therapy is a smart strategy to test and pursue further. And for this, we are now moving ahead with a nice grant from the EU. We collaborate with a company in the Netherlands, Intravac, that has experience with conjugate vaccines in the infectious disease space. And now we got funding to set up the GMP production of our antigen, do toxicology study, and then hopefully confirm efficacy of the final drug product in our mouse model. So if you are ERC funded and have something that's translational relevant, then I highly recommend this grant. We were in the first funding round, but they do this every year now, I think multiple times. So this was a really nice experience. On the team, we also have several neurologists from the DZ in Munich that, based on this data, will hopefully be able to come up with a nice clinical trial application so that we essentially can test that later in patients, although we need then more money for this. In parallel, we are also developing the monoclonal antibody for therapy, but here we have no in vivo data yet, apart from PK studies. We are just starting this, actually probably next week or the week after, because we just received a batch of antibody where we did affinity maturation and fixing the liabilities to make this a potential drug. I think this is something that could also be interested for the Huntington community, because our antigen is highly repetitive. And if you are thinking about making antibodies against these other reading frames, these problems could also affect you. So I show you some kind of nerdy primary data. So there are multiple ways to measure the affinity of an antibody. Classically, you code the antigen in this biocore assay on this chip, and then you come with your antibody as the analyte. Here, if we do this with Fab fragments of the antibody, we get extremely poor kinetics, so the antibody comes off quickly again. If we use full antibodies, then we get kind of a nice apparent affinity, but obviously there are avidity effects. If we do it the other way around, that we actually coat the antibody and use the peptide as an analyte, then we get relatively low affinities of only about 100 to 400 nanomolar. But this is also affected by problems of that this analyte is now aggregating. So this makes it also difficult to analyze. Then we did a third assay where we essentially did an ELISA and diluted the antibody and kept a low amount of the antigen coated on the plate. And here we get yet another value for the EC50 of this antibody. So I think probably the most relevant number is these biological assays in cell lines that I showed you previously, where we end up with about 5 nanomolar, so maybe in this range here, because of course we are using the complete antibody and not Fab fragments. Good. So this is kind of the first part where I hopefully convinced you that there's indeed one translation of the G4C2 repeat. PolyGA is the most abundant product of this one translation, and it can sequester proteasomes into these inclusions and impair proteostasis, which inhibits TDP-43 or promotes TDP-43 aggregation. And these monoclonal antibodies can inhibit transmission and downstream toxicity. Importantly, the active vaccination also can induce, with a proper conjugate, can induce striking immune responses that are beneficial, at least in the prevention setting, and based on biomarkers also in the therapeutic setting in these GA mice. In the last few minutes, I would like to show you something completely unrelated. also on this C9 pathway. Here we teamed up with Peter Heutink in Tübingen to look for small molecules to inhibit essentially one translation. So this was our aim, to look at to find a specific one translation inhibitor. And our top hit is decitabine, which is a chemotherapy drug, which in our hands has a really nice IC50 of about 50 nanomolar, and we replicated this in 4 different IPS lines, including one with rather short repeats. Here we get suppression. So the assay is based on measuring DPR levels using the MSD platform. Because the DPRs are so stable, we treated the cells actually twice and did a very elaborate protocol. So that's why we could only screen about 1,400 compounds, because this is a kind of expensive experiment. But at least we got one hit that works just as well as the ASO positive control. And then we looked further into this. Here we did repeated treatment over a longer period of time. As I told you that decitabine is used for chemotherapy, and it actually inhibits cell cycle by messing with DNA methyltransferases. But in the postmitotic neurons, it's absolutely non-toxic. So we can do this forever, and the cells live happily ever after, essentially. So we see that our compound is at 10 micromolar as potent as the ASO control to knock down or to reduce GA and GP levels. We tried really, we thought this would be a one translation inhibitor, but actually it's not. So I spare you the details, but we did a lot of experiments to test this hypothesis, but there's no effect on one translation. So then we looked at the RNA levels. C9 is a kind of complicated gene. So actually most of the transcripts are initiated at this exon 1B and don't contain the repeat. And about 5 to 10% of the transcripts initiate here at this exon 1A, and then the pre-mRNA contains this repeat, the expanded repeat. So we looked at these two transcripts individually using specific primers, and we can show that decitabine has, and also the ASO, has no effect on the transcript starting at exon 1B, because the ASO targets here, so this shouldn't do anything, and obviously our compound doesn't do anything if the transcript doesn't have the repeat. But the transcripts that do contain the repeat are actually suppressed by decitabine, and also 2 other compounds that have a less potency. And then we started looking at the RNA itself. So I had mentioned already that the repeat RNA is kind of coalescing into these foci in the nucleus in C9 patients, and also in this RPS model. And this can be nicely prevented by ASO therapy and also by decitabine for the sense foci. And then the reviewers made us look also at antisense foci, which the students didn't like at all, because it's a kind of annoying or complicated experiment. And but it actually is a quite nice result, because the ASO, as expected, had no effect on the antisense transcript, because it's a strand-specific ASO, obviously, while our compound, at least after prolonged treatment, nicely reduced the levels of these antisense foci also. So we are potentially better than the ASO therapy if we could make that into a safe, if we could convert it into a safe compound, essentially. So what we really so far couldn't solve is actually the mechanism of how all of this works at the molecular level. We excluded a couple of things, and one is also inspired by Huntington's disease, that repeats often are associated with DNA instability, and it would be imaginable that maybe DNA repair plays a role in this effect. But so we knocked down a couple of DNA repair factors in in the mismatch repair and base excision and nucleotide excision repair pathway, but they have no effect on the basal level of poly-GP or on the effect of decitabine treatment. So they are clearly not involved in that pathway. The positive control targeting C9 does suppress GP. We also looked at whether there's a kind of a repeat contraction. This has been also shown for some compounds in Huntington's. This is a little bit more difficult to analyze in C9 because the repeat is so much longer. But what we can show is that there is the treatment doesn't induce any really short repeats. So we can't exclude that the repeat is slightly shorter, but that's probably meaningless for the GP expression. But clearly there are no short repeats in this Assurogen assay. We also looked at the known target of decitabine is actually DNA methyltransferases, and other inhibitors against this target have no effect on C9 or DPR expression. Interestingly, our compounds have no effect on the wild type allele, so it's kind of an inbuilt allele specificity. So we think it works by acting on transcription of the repeat itself. We also have done a proof of concept study in mice where we did a really high dose of decitabine for 1 week. If we were to keep doing that, we would definitely kill the mice because it shuts down hematopoiesis essentially. But this led to a significant reduction in the sense foci and also DPR levels in the brain of these mice. So now we are trying to make this better because we accidentally, in Munich, there's a specialist for decitabine, and he gave us already some modified compounds, and he's making more for us. So we hope that we can dissociate the activity of decitabine or of these compounds between the toxic DNMT effect and our desired effect on repeat transcription. And then we're also trying to identify the target, and if we have that, we can screen for additional compounds inhibiting the target. So overall, we have a kind of a broad pipeline for C9. With decitabine and derivatives, we are trying to target sense and anti-strands transcription, which is upstream of the ASOs. Unfortunately, the Biogen one recently failed, and the wave formulation is still in the game. We are downstream of gene editing, obviously, and we are also targeting these poly-GA proteins because we think that that this could lead to a reduction in the disease mechanism, or not a disease mechanism, the existing pathology. So let's go back to the, let's go back to the Mission Impossible quickly. So I think I've convinced you that these are made. Actually, C9 is still a valid target. ASOs are in clinical trials already. We have found a specific inhibitor for transcription, not for the ribosome, but for transcription. We can link it to the TDP43 pathology. We made excellent antibodies. The active vaccination works in mice, and now I'm still stuck at this point. So I'm essentially looking for Tom Cruise to fix that situation. So if you are interested in doing a postdoc and want to be that guy, then please join my lab. I have to thank a lot of people, especially for the screen, Peter Heuting and Ashu. The immunotherapy is done by Chive and Nico. The screening is also was spearheaded by Mareike. Cryo-EM is with Ruben and Kiang. Thank you very much. I'm happy to take questions.
Okay, so thank you very much. This was very, very rich and inspiring. So questions?
Thank you very much. Very promising and exciting work. I'm wondering if you might comment on why a number of promising vaccine... Thank you very much. Very exciting work and promising. I'm wondering if you might comment on why various vaccine candidates have not worked. What are the sort of the steps where things might fail?
Yeah, I mean, definitely safety is an issue. I think with some, I mean, the Elan vaccine was a prime example where, which actually did reduce A beta, but it caused this meningoencephalitis. I think the tau vaccine by Axon Neuroscience is actually kind of promising. They, I think, also saw NFL effects. Although they didn't see effects on cognition in their phase 2 trial. I think that the advantage for C9 is that there is no endogenous function that we can kill with this. So that's why I'm a little bit more enthusiastic about this than other vaccine activities. But definitely, I think for the vaccine, definitely safety is the key issue, and getting a good antibody response in old people. But also the C9 people are younger than typical Alzheimer patients.
Thank you. More questions? Otherwise, we're perfect on time. So thank you very much. And
thank you.